Nanoparticle shape influences cellular uptake, but limited work has been done to change the shape of polymersomes. This protocol demonstrates how to elongate polymersomes for enhanced drug delivery applications.
This technique provides a simple approach that can create many different polymersome shapes with potential applications in drug delivery, pseudocells, or for other unexplored fields.
Elongated polymersomes can simultaneously deliver hydrophobic and hydrophilic drugs and more easily enter the cells. This technique could help shuttle drugs across the blood-brain barrier, addressing a major medical challenge.
To begin, dissolve 0.015 grams of selected polyester in 1 milliliter of DMSO by vortexing for 15 minutes and set up the solvent injection apparatus. Place a stir plate directly below the vertical syringe pump, then place a 5 milliliter glass vial with 1 milliliter of type 2 deionized water in a miniature stir bar on the stir plate. Adjust the syringe pump's height to allow for the tip of the needle to be fully immersed in type 2 deionized water, and set the infusion rate of the syringe pump to 5 microliters per minute. Perform the solvent injection by drawing the organic solvent and polyester solution into a 27-gauge needle. Place the needle into the syringe pump and make sure that it is secure. Adjust the pusher block to touch the syringe plunger's end. Start the stir plate so that the water is spinning at 100 revolutions per minute, and then start the syringe pump. Once the syringe pump has fully infused the organic solvent and polymer into the water, remove the stir bar and cap the glass vial. For characterization